Hello, my dear student. Assalamu alaikum. Hope you all are quite okay and fine. Welcome you all to my today online class. Before going to start my today's lecture, first of all, I would like to introduce myself to all of you. I am Mohammad Ghulam Morshed, lecturer of biology, Milestone College, Uttara, Dhaka. Today I am going to discuss about chapter 10, that is defense system of human body, immunity. The topics. Today I am going to discuss about the structure of antibody. I will be discussed here the type of antibody. I will also be discussed here the functions of antibody. Now the learning objectives. After completing my today's lesson, student could able to define some important definition like antibody, antigen, paratoph, hapten, epitope. You can also explain the different types of antibody. You can also explain the functions of antibody. Now, first of all, I would like to discuss about the definition of antigen. Antigen. Antigens means antibody generator. Anti means antibody, gen means generator. Just look at the a very simple definition of antigens. The substance which is recognized as foreign substance that when introduced into the body is capable of stimulating an immune response is called antigen. If you look at this definition, there is a new terminology, immune response. Immune response. Immune response, you should have a clear understanding regarding the immune response. When any foreign substance that when introduced into our body will stimulate the formation of immune response is called the antigen. Actually, antigen is responsible for stimulating an immune response. Now, antigens includes parts, cores, capsule, cell wall, flagella, fimbriae, and toxins of bacteria, virus, and other microorganisms. Most of the microorganisms act as an antigen, but there are some non-microbial agents are also act as an antigen. For example, insect venom, pollen grain, egg white. These are some non-microbial agents, but these are also act as an antigenic agent. Now the definition of epitope and hapten. This is the very important definition. Epitopes, the part of the antigen protein that is recognized by immune response, specifically by antibody, B cells or T cells, is known as antigenic determinant or epitope. So we can easily say that the epitope is the part of the antigen that is actually recognized by B cell, B lymphocyte, T cells or antibody is called the epitopes. Now the haptens. Some chemical molecules that react with a specific antibody but cannot induce the formation of antibody unless bounds to a special carrier protein or others large antigenic molecule. These are called the hapten. Actually, hapten is a very small molecular weight of lipid. Most of the very small molecular weight of lipid is act as a hapten, but these lipid molecules are not enough to stimulate the immune response, but when this small molecular weight of lipid combined with some carrier protein, then they becomes an antigenic agent. Now the definition of antibody. This is the very simple definition of antibody. Antibody is any of a large number of protein of higher molecular weight that are produced normally by specialized B cells after stimulation by antigens and act specifically against the antigens in an immune response. Actually, specialized B cells, specialized B cells, actually, as you know that the B lymphocyte is responsible for production of antibody. But how can a B lymphocyte can produce antibody? As you know that the B lymphocyte is originated from bone marrow, but maturation of B lymphocyte is also take places in the bone marrow. When B lymphocyte become matured, specific types of receptor protein will be developed, and when a specific antigen binds to a specific receptor protein of specific B lymphocyte, then B lymphocyte will be activated and undergoes repeated mitosis division to produce numerous clone B lymphocytes. This is called the clonal expansion, and this some of the activated B lymphocytes becomes plasma cell, and some of the activated B lymphocytes become the memory cell. This plasma cells is actually responsible for production of specific types of antibody. Actually, antibodies are a large molecules of glycoprotein which secreted from the B lymphocytes or plasma cells or B cells. Human body able to produce 100 million type of antibody. 100 million type of antibody. And each antibody is specifically binds to a specific antigen. Now let's discuss about the differences between antigen and antibody. Antigens. Antigens is a foreign substance that causes the immune response to produce antibody against it. But antibody is a protein that is produced by the immune systems when it detect a harmful substance antigens to neutralize or fight it. Antigens are made of either polysaccharides or proteins or lipid, but antibody are made of organic structural protein unit. Now, the antigens can be cells, but antibody are never cells. Antigens act as a key, antibody act as a lock. Antigens are typically divided into self and non-self antigens, but antibodies are there are total of 5 types of antibodies like IgA, IgE, IgG, IgM, and IgD. Now let's discuss about a very important topic, that is structure of antibody. Structure of antibody. Actually, look at the figure here. This is the structure of antibody. Antibody is a Y-shaped structure. I will be discussed here in details about the structure of the antibody. Structure of antibody. Each antibody is an immunoglobulin monomer. The Ig monomer is a Y-shaped molecule that consists of 4 polypeptide chain. If you look at the figure in the last slide, you can see there actually Y-shaped molecule structure. Ig immunoglobulin monomer is a Y-shaped molecules, and it is consisting of 4 polypeptide chain actually. 2 identical large heavy chains and 2 identical small light chains connected by disulfide bond. Structure of antibody. Each chain is actually composed of structural domain called immunoglobulin domain. Heavy chains with one variable domain followed by a constant domain, a hinge region, and two more constant domain. Light chains with one variable and one constant domain. The light chain contains about 200 amino acids, whereas heavy chains contains about 440 amino acids. The rod shape base of the Y-shaped antibody consists only constant domain of heavy chains. This region is called the fragments of crystallization regions or FC region. Each of the two expanded arm of the Y-shaped antibody consists of two heavy and light chain. This region is called fragments of antigen binding regions or FAB regions, which contains both variable and constant domain of each polypeptide chain. The variable includes the end of the light and heavy chains, which is the antigen binding site. The antigen binding sites of antibody is known as paratope. Actually, the variable regions of each light and heavy chains actually form the antigen binding site, and this site is called the paratopes. And the paratopes is actually binds to the epitopes of the antigen. Due to recombination of amino acid in paratopes through variable diversity joining, there will be 100 million types of antibody produced in human body. Now, let's discuss about the types of antibody. In human blood serum, there are 5 types of antibodies or immunoglobulin molecules. The types of antibody generally determined by the sequence of the amino acid in heavy chains of antibody. There are 5 types of heavy chains in human antibody based on the sequence of the amino acid. These are gamma chain, mu chains, alpha chains, epsilon chain, and delta chains. Accordingly, the antibody of human immune systems are classified into following 5 classes. Number 1, the immunoglobulin G or IgG. This is a general types of antibody with gamma chain. This is the most abundant classes of antibody in the body and constituting approximately 75% of the total, and is plenty in blood and lymph. Actually, IgG is the most abundant type of antibody. More than or approximately 75% of the total antibody is the actually immunoglobulin G, and these are mainly found in the blood and the lymph. It is a kind of antibody that works efficiently to coat microbes, spreading their uptake by other cells of the immune system. These classes of antibody can only transfer from mothers to fetus through placenta. Immunoglobulin M or IgM. IgM antibody with mu chains and remain attached to B cells. IgM constitutes about 5 to 10% of all antibodies in body, in blood. It is very effective in killing virus and bacteria. It is the largest Ig, which is the first secreted by the plasma cells. Actually, IgM is a pentameric form. Now, the IgA or IgA, immunoglobulin A, it is an antibody with alpha chain concentrate in body fluid, tears, saliva, and secretion of the respiratory and digestive tract, guarding the entrance of the microbes to the body. It is the second most abundant classes, constitute about 10 to 15% of the antibody of the serum. If IgA found in colostrum with huge amount. Immunoglobulin E. It is a rare types of antibody with epsilon chain and constituting about 0.1% of the total antibody in the blood. These types of antibody is actually located on mast cells and basophil. It influences releasing histamine with active the response of the inflammation. It is involved in allergic and hypersensitivity reactions and provide protection against parasite. Now, let's discuss about the immunoglobulin D or IgD. It is an antibody with delta chains and constituting about 0.2% of the all antibodies in blood. These types of antibody found in blood, lymph and B cells and plays a key role in initiating early B cell reception response. Look at the figure here. These are the different types of antibody. IgG is actually monomeric form. IgE also monomeric. IgD is also monomeric. But in case of IgM, these types of antibody is actually pentameric form. But IgA may be monomeric, dimeric or trimeric form. Now let's discuss about the functions of the antibody. Antibody-antigen reaction. Actually, antigen and antibody reaction actually occur when an antigen combined with corresponding antibody to produce an immune complex is called antigen-antibody reaction. Antigen-antibody reaction is occurred between the interaction between antibody and antigens. Functions of the antibody-antigens reactions are each antibody has a unique binding site called paratops. I have already discussed about the paratops in the whenever I discussed about the structure of the antibody. So the paratops which act as a locks onto the specific shape called epitopes of antigens. Antibody destroy the antigens which is then engulfed and destroyed by macrophage. Antigen-antibody reactions are of following types. Agglutination. Aggregation of microbes or cells due to antibody bindings is known as agglutination. IgM is responsible for agglutinin antibody. Opsonization. The antibody that is making the antigen susceptible for phagocytosis is known as opsonin. Sometimes phagocytosis process is not enough, or macrophage or neutrophils are not enough to engulf engulf the foreign particles of bacteria efficiently. But sometimes some antibodies which is called the opsonin, these types of antibody is actually induce or increase the susceptibility of the phagocytosis process, or induce the increased susceptibility of the antigens becomes more susceptible to the phagocytosis process, and these types of antibodies actually makes the antigen to becomes more susceptible to phagocytosis process. Now the activation of complement system. Antibody binding with antigen is not sufficient to remove the antigen from body. Then antigen-antibody complex initiate the activation and bindings of complement systems to it. And this is actually called the complement fixation. The lysis, complement activation can result in lysis of bacteria. Thus, complement fixing antibody IgM may indirectly causes lysis of susceptible target. Neutralization. Neutralization refers to the ability of antibody to block the site on bacteria or virus that they use to enter their target cell. Sometimes some antibodies can bind the bacteria in whose site that is actually used for the attachment of the host cell. Now the homework, write down the functions of antibody. The creative question, Dr. Tofazul said there is a Y-shaped protein substance in human body. He added that that protein substance keeps the body free from the certain disease. A. What is antibody? What is meant by paratops? Explain the structure of the mentioned substance. we analyze the last sentence of the Dr. Tofajjal.
Thank you for your passion sharing. I think today's lecture was very much effective lecture for all of us. I am humble requesting you all to watch my video lecture for several times so that you can understand each and every topics that have already discussed here today. If you have any queries regarding my today's lecture, feel free to ask me, or you can also ask your respective subject teacher. Thank you all. Khuda Hafiz.